Thank you very much, Keith. Yes, we are running a bit behind, and as my main job is timekeeper, I'm going to keep my comments to less than a minute. I want to welcome everybody. Thanks for attending. This is the first session on gene therapy. We're going to hold questions until the end, I have altered the times of the talks to the shorter of the two times everybody was told, 25 minutes and then 12 minutes for the second talks. I'm going to interrupt after 25 minutes. The questions, if we have time for discussion, should be submitted by chat, either by Zoom chat directly or by the conference chat, and the coordinators will forward that to Zoom chat. And please don't mention names of people or individuals or patients. So with those. So my very brief comments, which I'm no longer going to make, I was just going to try to distinguish between two senses of disease therapies based on genetics. As a geneticist, I kind of feel the need to do this. I'm just going to show the slide. There's the broad sense, which is genetic based, which is based on the knowledge of the molecular genetic basis of a disorder, covers all kinds of drugs, including gene drugs and a variety of delivery systems. And then there's the narrow sense, the gene therapy, which is delivery or alteration of the genetic material itself. This can be done in a variety of different ways. Much of it is under development. How it's done depends on the application. Delivery is typically via viruses or cells, but it can also be naked, like sort of with the Pfizer or Moderna COVID vaccines. So with that sort of as a very, very, very brief and broad perspective, I thought it might be helpful for people to hear about, because genetics permeates both of these. Just to get right into the session, I want to introduce the keynote speaker, Dr. Ben Kleinstiver from Mass General Harvard Medical School, who's our keynote speaker. He'll be giving us a broad overview on gene therapies, including CRISPR, and this will be followed by two shorter talks from other folks that I'll introduce at that time. And again, we'll hold questions to the end. And my clock still says 11:35, so I've been a good boy. Okay, I'll turn it over to the first speaker, Dr. Kleinstiver.
All righty, thank you. Thanks for the invitation to speak in this virtual conference, and I will do my best to stick to my allocated time. Yeah, I've been tasked with talking about the development of CRISPR genetic medicines, and I'll talk generally about that first, about our field first, and then get a little more into the weeds of what that actually looks like and how we begin to even think about that. But as said, my name's Ben Kleinstiver. I run a research lab at Mass General Hospital and Harvard Medical School in Boston. I'm originally from Canada. I grew up near Toronto, did all my school in Canada, and then moved to Boston right around when CRISPR was becoming, the new genome editing technology. And I'll tell you a bit more about, why CRISPR is so exciting and all the really amazing things that we can do with these new tools to modify genetic sequences. As with any talk, my institution requires me to just show you that I do have some potential conflicts of interest, which I usually refer to just as outside activities. I consult and advise for various companies and obviously file patents on the work that we do, only a little bit of which I will tell you about today. I think I only have 3 data slides in the presentation. Okay, so as background, just about myself and the types of things that we work on in my lab, we're really interested in developing genetic medicines. And to do that, we have to work on lots of different topics, because this is a really broad field that relies on us building best-in-class technologies. So as I'll tell you more about today, essentially these 3 themes, what are genome editing technologies? How can we make them more optimal? I'm a biochemist by training, so we think about these molecules as proteins, because this is what they are, and how we can use a method called protein engineering to make them have more desirable properties, to make them edit safer and more effectively at the DNA site of interest to correct a genetic change. And then also, we're trying to use CRISPR to treat different genetic diseases. So I'll talk really briefly about that and just other exciting things that are happening in the field. But altogether, hopefully this will give you a quick overview about what's happening in the genome editing space and how we can then think about applying these technologies to correct different diseases. Okay, so I'm not a geneticist, but I felt compelled to put in a basic slide to talk about, why diseases arise. And, for individuals who don't have known genetic diseases, we presume that we have, a wild type genetic sequence. And this just means that we don't have known pathogenic alterations in our genetic code in our cells. But most people that have a genetic disease have these spelling mistakes or errors in their sequences of their cells. And this can look different depending on the individual, but in general, these are mostly single nucleotide changes. So, of our 3 billion bases of our genetic code in our cells, single changes can actually make a really big difference as we think about disease. Of course, there are lots of different types of substitutions, and I'll get into that, but it's really important to emphasize here that these really small changes can cause disease. Another really large class of disease is caused by what's called insertion or deletion mutations. So this is essentially adding in genetic sequence or removing genetic sequence that changes essentially the cadence of how we read our genetic code. And that can cause problems when you're trying to translate a protein sequence from DNA to RNA to then to protein. So these insertion or deletion mutations really not only impact the location of the mutation, but also the genetic information downstream. And then, although this is a slightly smaller class of genetic disease, this is still very common, larger structural changes to our genome. So this could be things like chromosomal changes, loss of an entire gene, or even duplication of a gene, where sometimes the exact amount of a protein that you make in a cell is actually quite important. So as we think about known pathogenic human genetic variants, this is, I guess, 3 years old at this point, but probably categorically still true. Most human diseases are caused by these single nucleotide changes here. So these, single base changes. Many diseases, over a quarter are caused by these insertion or deletion mutations, and then some other fraction of the remaining diseases are caused by these larger genetic changes. So I mentioned this just to give you a sense of what we're contending with as we think about developing genetic medicines. We can't develop one tool to try and treat all these different types of genetic diseases because at their core, they're very different in terms of what they look like and how we would need to correct them. Okay, so for the sake of time, I'm not going to belabor this, but as we think about traditional medicines, really this is small molecules or antibodies that really don't address the underlying genetic cause of the disease. They can treat symptoms, they can try and ameliorate some sort of dominant negative protein, but really even as you take these medicines, you still have this genetic mutation in your cells. So the fields of gene therapy and gene editing really try to overcome that at the genetic level. Although I have never participated in any kind of gene therapy research, a lot of my colleagues in the center that I work in definitely focus on gene therapies, but I see gene therapy and gene editing as complementary for reasons that you'll see. So gene therapy really is taking a healthy copy or a normal copy of a gene, putting that into some sort of delivery vector and then delivering that into a patient. And the hope there is that by delivering a normal copy of a gene, you can compensate for the mutant copy that exists within that patient. This doesn't actually fix the underlying genetic cause of the disease, but just hopefully compensates by providing this normal copy of the gene. So although gene therapy has been really successful for many different diseases, I think the point here that I want to make is that the underlying genetic mutation still hasn't been addressed in patients that are treated with gene therapies. So that's really where the power of gene editing has come to bear on patients and for academic research. We now have these tools, lots of different analogies, tweezers, a scalpel, all kinds of different ways that you can think about precision genome editing, where we can now change the underlying genetic material in a cell. And as I showed you a few slides ago, that looks a lot different depending on the gene and the mutation. But really the point here is to say that we can make these changes now to the genetic code of life in cells of, in a petri dish in the lab, or in an organism like a mouse or a human. So I've got a few minutes where I'll try and quickly just walk you through three different topics. I'll tell you about some of the really exciting technologies that we have in the genome editing toolbox. I'll talk really briefly about some of the properties of these CRISPR tools that we think about, and this is where my lab has done a lot of work to try and make enzymes that are safe and effective as therapies. And then, time permitting, I'll talk a little bit about some of the really exciting things happening in the gene editing field in terms of developing genetic medicines. Okay, so I hope this doesn't get too much into the weeds, but really a CRISPR enzyme, you can think of this as a protein in a cell. It interacts with the DNA sequence, and the job of that CRISPR enzyme is to break that DNA sequence into two pieces. And you might be thinking, well, can't that also cause a disease, right? If you break DNA and it goes back together the wrong way, and the answer is yes. But by manipulating this process and by breaking DNA in a specific spot, this also gives us the opportunity to repair that sequence by convincing the cell to repair it in a certain way. So again, at the core, this is how gene editing technologies work. For the most part, the version 1 technologies, which are called nucleases, they break the DNA intentionally, and that allows us to make changes at that site of the DNA break. So what happens when we break DNA is that the biology of the cell, these natural DNA repair processes take over, and they try and actually put that DNA back together. And if the cell makes a mistake while it does that, it can create these mutations that I'm trying to illustrate here. So insertions or deletions or changes at that break site. And that's, as researchers, a tool that we can use to knock out the function of a gene, which essentially is what many mutations look like in patients. So although this strategy here is generally not useful for correcting disease-causing mutations, it does give us a tool to study the biology of genes in ways that we can then leverage that to make different types of corrections. Another way that a cell can repair a DNA break is if we provide a sequence that break can then use to repair against, so what we call a donor DNA molecule, encoding the exact change that we want to place into that genetic region. That can lead to precise changes. So we break the DNA and then we provide a copy that's correct that can then be used as a template to correct that break. And that allows us to make really precise changes to the genome. So you could imagine how we could then use this to correct disease-causing mutations simply by providing the normal sequence in this donor DNA molecule. So these types of approaches have existed a long time, even pre-CRISPR, with other genome editing technologies, essentially since the early 2000s and even before that. And these basic methods of editing genes is really to knock out the function of a gene to study the biology, or to make a correction to try and reverse that sequence back to the normal wild type composition of that sequence. So those are what we like to think of as first generation CRISPR technologies. But fortunately, in the field, we've had all these amazing advances in the last few years that give us the ability to make more precise edits to the genome. So here, this graph taken from Alexis Comore's paper back in 2015 was just to break down the types of the single genetic mutations that cause disease. And what David Liu's lab, then at Harvard, now at the Broad Institute, wondered is if they could create enzymes that essentially reverse these single nucleotide changes. And I won't, again, try to get too into the weeds here, but we can essentially take a CRISPR enzyme, target it to a site in the genome, and attach what's called a DNA deaminase domain to that enzyme. And this enzyme performs precision chemistry on the bases where you target that CRISPR enzyme, meaning that we can make single letter edits to the genome with these base editors. That's why they're called base editors. So this first generation was called a C to T base editor, and that allowed us to take the CRISPR enzyme decorated with this catalytic domain that performs chemistry on these DNA bases that allowed us to make these C to T edits. a few years later, they developed another complementary tool called an A to G base editor that allows us to make A to G edits. So base editors really are super powerful when you think about the types of changes we can now make to the genome. Instead of breaking the DNA and relying on the cell to put it back together in the way that we want it to, which doesn't always happen, we have these really precise enzymes now that we can target to a specific spot in the 3 billion bases of our genome, and then have that enzyme perform this really precise single base chemistry to make C to T edits or A to G edits. We're really excited about the potential of base editors, and obviously many others as well, to make these single nucleotide edits in cells of patients or in organisms. Okay, so despite all that promise with base editors, I think there are still some types of genetic changes that base editors can't make. So recently there was another new version of CRISPR enzymes that were developed called prime editors. I won't get really into the details here, other than to say that this allows us to provide a genetic sequence also into the cell, and you can decorate the Cas enzyme with some other functional domain now to write that sequence into a genetic locus. So we now have the power to make really small changes, not just single base changes, but multi-base changes using these tools called prime editors. And I'm happy to expand more on this if people have questions later. Okay, so as we think about why CRISPR has been so amazing and allows us to make these changes to the genome, really it's just that we can use this molecule called a guide RNA. So this is a short RNA molecule, and we can program the Cas9 enzyme with this molecule, which essentially tells it where to go in the genome. So the Cas enzyme, when it's bound to this guide, it searches 3 billion bases of sequence for the 20 nucleotides that match this part of the guide RNA. So this is why CRISPR is so powerful, because we can tell it where to go in the genome and make these specific edits. And again, if we think about all these categories of genetic mutations that cause disease, as I told you on the last few slides, we actually now have many different tools that allow us to address essentially almost every piece of the pie here in terms of the types of mutations that cause disease. So to summarize those, we have version 1 or generation 1 CRISPR that allows us to knock out genes, which, if you have what's called a dominant negative mutation, this is one way that you can think about treating that sequence, is by just knocking it out. We have base editors that allow us to correct single base changes in the genome, which is, over 50% of pathogenic mutations are single base changes. We have these newer tools called prime editors that allow us to write specific sequences into the genome, which allow us to not only address single base changes, but also other, small scale, but non-single base change level edits. And we also have CRISPR enzymes that allow us to regulate gene expression, either at the DNA level or at the RNA level. So we can perturb the expression of genes in different ways to, study their biology, but also think about knocking them down therapeutically. And I should say that this is like a real quick generalization. We have lots of other tools too, but I'm trying to focus on the ones that I think are, most appealing as technologies. Okay, so here, lots of words and funny pictures and jargon, but I think it's important to make the point that the genome editing field did exist before CRISPR. We and others had been working on different versions of technologies prior to CRISPR being developed as a genome editing tool in 2012 and 2013. The problem with these tools is that they're all protein, and to convince a protein to bind a different sequence in your genome is actually very challenging to do. It takes a lot of time and it's very expensive. Convincing a protein to read a different DNA sequence is not trivial. On the other hand, CRISPR, as I told you, we can program to bind new DNA sequences by simply changing its guide RNA. So here we just have to change the RNA sequence instead of the protein, which is fundamentally much easier. We can synthesize a new guide RNA for a matter of dollars instead of spending 6 months or $30,000 to reprogram all these prior technologies. So this is where CRISPR has been so transformative for genome editing, is that we can make edits now at different spots in the genome by reprogramming an RNA molecule instead of having to extensively re-engineer the entire enzyme. Okay, so hopefully that gives you a bit of a sense of the flavors of genome editing tools that we have to make different perturbations to DNA to correct mutations and whatnot. And really the ability of CRISPR to edit the genome is amazing in terms of what we can now do. And that's really because we can generate these guide RNAs inexpensively and rapidly. Okay, I will just talk briefly about the properties of genome editing technologies and how we in my lab think about building new versions of enzymes that have new functions. So if you think about a genetic medicine, you'd want this genetic medicine to be highly active so that you edit all cells in that patient. You'd want that medicine to be able to make any edit that you want. So being able to access the entire genome. And you'd also want that medicine to be really safe, right? Making the edit only at the site of interest that you want to make that edit and no other parts in the genome. One of the issues, my slides will advance, with CRISPR is that it actually comes from a bacterial immune system. So without getting too deep into the details here, you know, CRISPR evolved as a way for bacteria to defend themselves against viruses or bacteriophage or other types of DNA. It did not evolve for us as humans to use that to edit DNA sequences. So that's where we can use new methods in the lab to make versions of CRISPR enzymes that are safer and more versatile. One of the properties of a CRISPR enzyme that's really important to think about is where can it actually bind in the genome? And there's this short sequence called a PAM that the Cas9 enzyme has to read to be able to sit there and then make an edit to the genome. And practically what this means for genome editing is that you can actually make edits only where we find these PAM sequences. And then the regions in between that don't have PAMs, these CRISPR enzymes can't efficiently bind and make edits. So as we think about all these precise changes to the genome that we want to make, the availability of these PAM sequences are really important. So we've done a lot of work over the last few years to build new versions of CRISPR enzymes that can read new genetic sequences. Either by modifying parts of the protein to make it read new sequences, or essentially removing this part of the protein to allow it to read the entire genome. For the sake of time, I won't tell you how we did that, other than to say that now with all these new tools, we can read and edit the entire genome. So that is no longer really a limitation for us as a field. And virtually any mutation now that occurs in a patient, we do have technologies that should allow us to at least target a CRISPR enzyme near that mutation. Another really important property is the safety. And I think this, you know, as we think about genetic medicines or any type of medicine in general, the safety of a platform really is at the top of our minds. And as we think about a genetic medicine, we want the CRISPR enzyme to edit only at the site of the genome that we want, but not other parts of the genome. Unfortunately, CRISPR enzymes, you know, because they're a bacterial immune system, they've evolved actually to tolerate mutations in the sequences that they edit, which means they can read, bind, and edit other sites in the genome that look a lot like the intended on-target site. So we as a field have developed lots of different methods to try and monitor whether or not these off-target edits happen. We've also developed lots of ways to try and mitigate these off-targets. I could spend 3 hours talking about all of this. But just briefly, I'll tell you a story about how a few years ago, and in continuing work in my lab, we've built these high specificity enzymes that no longer read off-target sites. So this is the second of 3 data slides that I will show. And even though I'll just walk quickly through this, this is an editing experiment in cells where we, with our CRISPR enzyme, get editing of 50% of the cells, well above the background error rate in the experiment. But with CRISPR enzymes, what we've found is that they also can edit these other off-target sites in other parts of the genome well above the error rate of our experiment. So what this means actually is that Cas9 is reading and editing other parts of the genome in ways that we really don't want it to. So these gray dots being above the red dots is not a good thing for the safety of genome editing. So what we did is we built a high fidelity version of this enzyme that had much better proofreading. So now in the blue dots here, you can see that the blue dots overlap the gray dots, meaning that this enzyme edits at the intended site with the same level of efficiency. But now at these other sites in the genome that we don't want to edit, you can see that the blue dots with this new version of the enzyme that we engineered are really at the same level of the error of our experiment, way below what we saw with the normal Cas9 enzyme that came from nature. So what we've been able to do here is preserve the intended edit, but now eliminate off-target edits. Practically what this means is that we can go from an enzyme that naturally has off-target effects, which again, as we think about the safety of a therapy, are an undesirable side effect, I guess we can think of. Now, by using these engineered versions of the enzyme, we can edit only at the site of interest. Okay, so I've got 2 minutes to talk about probably the most exciting stuff. What a genome editing medicine or therapy looks like. And some diseases we can treat by taking patients out of, or cells out of the patient and treating them what we call ex vivo in a dish, making these genetic corrections, expanding these cells, and then reinfusing them into the patients. But for most diseases, we don't have that luxury. It's impossible to take out some organs or cell types, edit them, and then deliver them back into the patient. So this is where, as a field, both for gene editing and gene therapy, we need these delivery vehicles like viruses or lipid nanoparticles that allow us to deliver these tools into the tissue of interest to make the correction. So as we think about any genetic disease, we sort of have this philosophy. What we do in the lab is we create a model of that disease so that we can scalably think about ways to correct, knock out, or edit that disease-causing sequence. The types of tools here, for the sake of time, I'll just skip through. This is just some data in the lab. We're working on different ex vivo therapies, but I'll just quickly mention some of the exciting things happening in the field with in vivo editing, because this, again, is one of the major barriers for genetic medicines, is how do we deliver these tools into the patient for different types of diseases. So here a couple of years ago, a company, Intellia Therapeutics, essentially published this paper showing preclinical data that you could deliver CRISPR enzymes using a lipid nanoparticle as a delivery vehicle into the liver of patients to edit cells in the liver. These lipid nanoparticles is essentially what most of the coronavirus vaccines are protected with, where you can be injected with that, and they mostly go to the liver for metabolism or for expression. So this was a really exciting result for the field, showing that we could edit the liver in vivo in a living animal for the first time. Here, this approach has then been continued by this company into clinical trials with human patients with this disease. And as of last year, these patients were doing well and had really high levels of editing in their livers, which in principle correct their underlying genetic disease. There are other ways that we can deliver into different organs. You know, for individuals that have deafness, we can think of different ways to deliver into parts of the ear as an organ to correct the underlying genetic defects in those types of cells. And then really excitingly, I guess last year, David Liu's lab published a paper with in vivo editing for progeria. And here this was essentially a systemic injection of a virus carrying the CRISPR enzymes that led to high levels of correction in this mouse model of the disease. But really excitingly, you know, the survival of these mice is normally a couple, I guess, a month and a half or so. But basically to the end point of this assay, more than a year later, these CRISPR-treated mice were still alive, living much, much longer than they would have without this CRISPR therapy. Okay, so our work really has focused on how do we scale the utility of these CRISPR enzymes so that we can make essentially any mutation addressable. Because I didn't have super much time, I didn't really get a chance to tell you all about that. But I'll just wrap this up by summarizing. You know, I started this talk by showing you all the different types of genetic mutations that cause disease. And really, I think we're in a fortunate point as a field to have tools for any of these given classes now to be able to actually think about correcting these genetic causes. For single base mutations, mostly we would think of using these tools called base editors because they allow us to make these precise single base changes. For these insertions or deletions, we can use CRISPR nucleases, either with or without a repair template, or prime editors to make these precise changes. And then for larger structural changes, we can use tools like prime editors or other types of enzymes, others in development, which I can expand on more later if needed, to make these large scale changes. And I think also as you think about a patient population where these single base mutations are not consistent between individuals. These larger approaches where we could introduce a corrective gene into the whole patient population obviously has a lot of attractiveness, so that we don't need to develop a bespoke therapy for every individual patient. Okay, so to summarize, I told you a little bit about some of the really new exciting genome editing technologies that exist in the field. I talked really quickly about some of the properties of CRISPR enzymes that, as we develop genetic medicines, we really have to think about to be able to maximize the efficiency of the editing and also the safety. And then just really briefly told you about some of the exciting things happening in terms of in vivo genome editing therapies in the field. So I'm happy to take questions now or later in the panel. I forget what the format of that would be, but in the meantime, just highlighting some of the folks in my lab that are doing some really amazing work that I didn't tell you much about today, but I'm happy to answer questions if anyone has them.
Thank you very much. That was fantastic.
Thank you.
I would have loved to have given you another half hour at least, let alone just the extra 5 to 10 minutes that we were all given. If you could unshare your slides, I would introduce the next speaker, and we'll take questions at the end. And I do want to give the organizers a chance to think about and let me know whether or not you are willing to extend our question session so we really have more time. Otherwise, I'm going to have to be really rude to the next two speakers. But thank you very much. That was fantastic. I'm not even going to share my slide. The next speaker is Phoebe Yan from Amy Ramsey's Lab at University of Toronto. We'll talk about AAV gene therapy trials I believe in GRIN1 and in GRIN2B patient-based genetically targeted mice. So take it away.
Okay, so thank you for the introduction. I hope everyone can hear me well and see the slides. Hello, everyone. My name is Phoebe. I'm a PhD candidate in Dr. Amy Ramsey's lab, and I'm currently working on the gene therapy projects in mind with GRIN1 patient variants. So the gene therapy approach we are using here refers to the delivery of a functional gene into the nucleus of the cell that can be expressed, but won't actually integrate into the genome. So as Dr. Kleinstiver said, it won't correct the mutation. The delivered gene will act as an extra copy of the functional gene to compensate for the gene loss or mutation. Therefore, we asked, would delivery of a functional gene GRIN1 gene be a long-lasting treatment for patients with GRIN1 variants? Several properties of GRIN1 has made this approach feasible. So first, GRIN1 encodes the subunit that is expressed in all functional MD receptors. Therefore, we won't be too worried to globally deliver GRIN1 gene to different cell types and slightly overexpress it. Another important thing about gene therapy is whether restoring the gene expression could improve symptoms that are already shown in the patients. Our lab has previously shown and published in Molecular Psychiatry in 2020 that the consequences in a mouse model with only 10% GRIN1 expression, which we call the GRIN1 knockdown mice, can actually be rescued in the adult mouse brain, therefore supporting the use of gene therapy to rescue the symptoms in patients with GRIN1 variants. So now let me explain a little bit more in detail why we think this approach might work. So in the cells with the heterozygous GRIN1 variant, although both mutant and wild type proteins get expressed, more than half of the proteins will receive at least one mutant subunit, which is enough to mess up with the entire receptor function. We propose to deliver wild type GRIN1 gene as an extra copy of the functional GRIN1 gene. We hope that with this extra copy, it could lead to expression of more wild type GRIN1 proteins in the cell, which could compete with the mutant proteins for the assembly into the MD receptor, so that more functional receptors can be expressed. In such a way, we don't need to actually correct the mutation, but the phenotypes can still be rescued. Therefore, in theory, regardless of what exact mutation the mice or the patients have, we can still treat them with the same intervention and have similar benefits. So to deliver the gene into the cell nucleus, we need a vector. And the type of vector we decided to use is called adeno-associated virus, or AAV, which are advancing tools that can be used for therapeutic gene delivery targeting the central nervous system. I want to point to a particular family of AAV, the AAV-HSC family, which refers to naturally occurring AAVs isolated from CD34 positive hematopoietic stem cells. So basically they're derived from human cells. This vector can enter the central nervous system and peripheral tissues following IV administration. And because they're derived from human cells, it also has the potential to have fewer side effects and better transduction results. So we're very grateful to have the opportunity to collaborate with Homology Medicines, Inc. with their expertise in the AAV-HSC works and our knowledge in growing disorder and animal behavior studies in the Ramsey Lab. We designed this comprehensive study plan that is separated into 3 phases. So the first phase is basically to design and produce the vector to be injected into the mice. Homology helped us to select a good AAV-HSC vector for our project. And then we designed a sequence containing the wild-type green DNA that we want to deliver, which we call the plasmids. We synthesized the plasmids last year and sent it back to Homology for them to package it into the virus to make the final product. Now we have the final product and we can move into our in vivo studies in the mice. So with Homology's experience in using AAV-HSC for in vivo works, they helped us design the route and dose for injection. And then later in collaboration with Dr. Ben Kleinstiver's lab from U of T, we will characterize the mice behaviors and AMD receptor functions in mouse lines with different GRI patient variants. And then the third phase is basically using the tissues from these mice to look at the expression of the delivered gene using both biochemical and histological techniques. So with the support of all our collaborators, we'll be able to address the following questions in this project. First, we'll be able to see where the delivered gene is expressed. This depends on 2 things. One is the vector that we selected. The one we selected has the ability to enter what we call transduce into the brain, but we also need to keep in mind that it also enters other tissues like the liver and heart. 2, the promoter, which is a sequence in front of the GRI gene we want to deliver. So different promoters drive gene expression in different cell types. We have 2 promoters here to test out. The GAPDH promoter drives gene expression in all cell types, while the SYN1 promoter drives expression only in neurons, which is the major cell population we want to target. The second question is, which mouse line should we treat? Since we hypothesized that our intervention has benefits regardless of the exact mutation, we would like to test it in as many GRI patient variant lines as possible. In collaboration with Dr. Kleinstiver, we will be able to test our intervention in at least 4 mouse lines, and including both loss of function and gain of function mutations. When to inject? As our first study, we decided to start the injection in young adult mice. The next question is where to inject. We proposed 2 routes to test in this project. First is intravenous, which is directly into their vein blood. This is the least invasive route. It benefits both the peripheral tissues and the central nervous cells expressing the AMD receptor, but might also cause problems in tissues that we don't want to target, for example, the liver. The second route is intrathecal, which is into the fluid space surrounding the spinal cord, called the cerebral spinal fluid. It is slightly more invasive, but it leads to more expression in the central nervous system cells with less peripheral side effects. But at the same time, the peripheral tissues actually have AMD receptor expression won't receive the treatment. How much to inject? We will try several doses and perform a dose response study to find the optimal dosage for this intervention. And as I mentioned in the previous slide, we will use behavior tests, functional assays to see what phenotypes are improved, and also use biochemical and histological experiments to visualize and quantify the expression of the delivered GRI gene. So the first thing we need to confirm is that can the AAV-HSC delivered GRI gene be expressed or not at all, and where they're expressed. So this leads to our study 1. We start with the sequence with the GAPDH promoter for expression in more cell types. The mouse line we used here is a heterozygous GRI1 Q536R mice, which is thought to be a loss of function mutation at the glycine binding site. We injected the mice at postnatal week 8, meaning 8 weeks after they were born, the young adult mice I mentioned, through intravenous injection. Because our main purpose is to see the expression, so we used a relatively high dose challenge here to get as much expression as possible. We allowed the viral vector to be in life for 4 weeks for the delivered gene to reach a stable expression level. Then at the age of postnatal week 12, we performed some behavioral tests for exploratory purpose. So my colleagues have done a thorough behavior study on the Q536R mice, and they saw some phenotypes in the visual cliff and puzzle box. We want to see if the gene therapy could actually improve those. And although we didn't see a phenotype in the wire hand test, but we still included it, and I'll explain later in more details later. So after that, we collected tissues and for the main purpose of study 1, which is to look at the expression of the delivered gene, and we can do that in different levels. So the VGS or vector genome counts basically looks at where does the virus go. The qPCR looks at the green expression at the mRNA level, and then Western blot and IHC could look at GRI1 expression at the protein level. So we will have a more comprehensive idea of where and how much GRI1 gene is expressed. So the behavior tests are only exploratory, but I would still like to share with you this very preliminary results. But I do want to clarify that since we only have a small number of mice in study 1, therefore none of the results I'm talking about here are statistically significant. So the first test we did is called visual cliff, which looks at the visual depth in the mice. For the sake of time, I won't explain in detail how what this test does, but basically a mouse with intact visual depths, for example, the wild type mice shown in black here, will spend more time in the safe area comparing to the cliff area, which is also shown here by the discrimination ratio. Like most of the wild types have a discrimination ratio larger than 1, meaning they spend more time in the safe area. The Q536R mice, however, spend similar times in the cliff and the safe area, with the average discrimination ratio slightly lower than 1. The Q536R mice receiving the treatment, however, they actually look more like the wild types, indicating the visual cliff behavior seems to be rescued, and they have a discrimination ratio larger than 1. But again, none of these results are actually significant. So the next we did is puzzle box, which looks at the executive function and problem solving in the mice. So we basically put the mice in the starting box, and the goal is for them to enter the goal box, and we count the time for them to get in. And we gradually make the task harder for them on day 2 and day 3 of the experiment. So as you can see, the Q536R mice spent more time comparing to wild types to get into the goal box, especially on day 2 and day 3, when the tasks are more difficult. While the mice receiving the treatment seems to be somewhere in between, indicating some improvements in their puzzle box behavior, but with these huge error bars, none of the results are significant at the moment. We also performed the wire hand test, which looks at the muscle strength and motor function of the mice. We actually did not see a baseline difference between the wild type and the Q536R mice as shown here, which is consistent with our previous observation. However, the mice received the treatment seems to fall faster with a weaker muscle strength. But again, the results are not significant. Also, since we are giving a high dose challenge here in study 1, it is not too surprising to us that we might see some treatment related side effects. So up to now, we have completed the injection and behavioral characterizations for mice in study 1, and we have collected their tissues and currently working on the biochemical and histological experiments, which is to see where is the AAV-HSC delivered GRI1 gene expressed and to what level. So from this preliminary results, we saw some promising trends of the behavioral improvements in adult GRI1 Q536R mice receiving the AAV delivered wild type GRI1 gene. However, we need to increase the number of mice to determine if these improvement trends are actually real. And we also need to see if these improvements are actually associated with the delivered gene expression, which is what we are currently working on. And after we're done with study 1, we will start optimizing our intervention. Potential changes we can make include changing the vector to get better transduction and expression profile, find a better dosage and route of administration. So using the optimized intervention, we'll perform a comprehensive phenotypic characterization in mouse lines with different GRI patient variants. We won't only focus on the behavioral tests where we saw a phenotype, we will actually perform a full spectrum behavioral study so that if there are any potential treatment-related side effects like the wear hand that we saw, we can capture them in early stage. So with that wraps up my presentation for this exciting project, and I'll keep working on it. I would like to thank everyone in the Ramsey Lab who has helped with the project, and the great scientists from homology who also made significant contribution to this project. I'd like also to thank all our collaborators from other labs and our funding sources. And finally, thank you again for your continuous support and holding this amazing event.
Thank you very much. That was really great. Some essentially promising results. And thank you for almost sticking to the 12 minutes. The last talk, we've been given a little bit more time for discussion, but we still need to try to keep to the 12 minutes. I apologize again for that. Last talk of the session is from Joe Anderson from UC Davis. He's the Associate Director for the Gene Therapy Center. So he knows a lot about gene therapy, and he'll talk about work on Angelman syndrome, stem cell gene therapy, and the potential for GRI disorders. Dr. Anderson, thank you.
Hi, everyone. My name is Joe Anderson. Thank you for inviting me to present on our work. I'm going to talk to you today about a hematopoietic stem cell gene therapy approach for GRIN disorders and the potential for this therapy. The approach that we're taking is based off of a method called cross-correction. This has been used for lysosomal storage disorders, leukodystrophies, and my lab has recently used this approach to treat Angelman syndrome in the mouse model of Angelman syndrome. And so what we're now doing is trying to apply this approach to other neurodevelopmental disorders, including GRIN. And so we were approached by the GRIN Foundation to evaluate this approach for GRIN disorders. So the way that this works is that you have a donor cell that is expressing the wild type and functional copy. Ben had touched on that, and then Phoebe just talked about that using AAV. But instead of using an AAV approach where we would be directly targeting neurons, we're actually going to be using the hematopoietic system and blood cells to express the functional and wild type copy of GRIN genes, and then allowing those cells to transfer them to neurons in the CNS. So as I mentioned, we've demonstrated that this approach works for Angelman syndrome in an animal model, both in preventing and rescuing Angelman syndrome phenotypes. This work was recently published in Human Molecular Genetics in 2021, and it's freely available for anyone to go check it out. But what we showed is that we were able to prevent and rescue phenotypes in an open field locomotion, beam walking, gait correction, novel object recognition for memory and learning. And then also we were able to decrease the EEG delta wave, which is this delta wave is increased in its spike level in Angelman syndrome patients. So we were actually able to decrease this. So the way that this approach would work is an insertional gene therapy approach, where we would take the wild type and functional copies of GRIN genes, express them in a vector system, and then insert them into our cells of interest, which will be the cells of the hematopoietic system. And then these cells will express this functional copy of the GRIN genes and then transfer them to neurons. The vector system that we're using, unlike the AAV system, is called a lentiviral vector system. And the reason why we chose this vector system is that the GRIN genes that we want to insert into the hematopoietic cells will be inserted into the genome of the target cells for the rest of the life of those cells. They're permanently integrated. So then any cell that's derived from those cells, daughter cells, if the hematopoietic cell divides, those new cells will actually get the copies of the GRIN genes that we inserted into the original cells because they're permanently integrated into the DNA. The reason why we wanted to use the hematopoietic system is because the blood cells and the blood system is found throughout the body, including in the CNS. There are glial cells called microglia cells that are from the hematopoietic system, and they reside in the brain and in the CNS. And so we want to use these cells to transfer these wild-type GRIN proteins to the affected neurons. This is a classic hematopoiesis tree. And so you can see way at the top, it's the pluripotent stem cell. And that's the cell that we're going to be targeting, the CD34 positive hematopoietic stem cell. It resides in the bone marrow of everyone, and it constantly makes white blood cells and red blood cells for the life of us and any patient that we would treat with this therapy. And so if we're able to target this stem cell, there's a potential for a one-time treatment because it would happen like a normal bone marrow transplant where we would target the stem cells and then transplant them into the patient, and they would make all these immune system cells, including the microglia cells in the CNS for the life of the patient. So the way this would happen is we have blood stem cells, we have a GRIN expressing lentivector. We transduce those blood stem cells with this vector that expresses these wild type and functional GRIN genes, and then we get GRIN expressing immune cells. So we are collaborating with Dr. Amy Ramsey and Graham Collinridge, and we're initially going to use GRIN1 and GRIN2A genes, looking at different combinations and modifications of these genes. And so my lab will be making the constructs, making the vectors, and then we will send them off to Drs. Ramsey and Collinridge so they can evaluate the mice in a similar way as Phoebe talked about with some of her behavioral studies. So this is how the research would go. We would collect bone marrow from donor GRIN mice. We would transduce this bone marrow with the vectors. We'll then use adult GRIN mice. They'll get busulfan conditioning, and this will make space in the mice and in the bone marrow for our transduced donor bone marrow cells to engraft. These cells can be injected intravenously, and the bone marrow stem cells know where to home back into the bone marrow, and then about 6 to 8 weeks later to let the cells engraft, differentiate into immune cells, and then also engraft into the CNS. Then we can start doing some of the behavioral and phenotypic assays to see if this is effective. As far as a future clinical trial, the way this would work is a normal bone marrow transplant will mobilize and isolate stem cells from a patient. It's just an intravenous draw similar to donating blood. We'll engineer the stem cells with the lentiviral vector that expresses the GRIN genes ex vivo, so outside of the body. A patient will get a conditioning regimen similar to the busulfan that we're going to use with the mice to make room in the bone marrow so these cells can engraft. And then the genetically modified stem cells will then be intravenously injected into the patient. The stem cells know to home back to the bone marrow, and then we will look and follow the patients for improvement. So I'd like to acknowledge the people in my lab that will be working on the vectors, and then also Drs. Ramsey and Collingridge, who will be evaluating these vectors in the mouse models, and then also the Cure Foundation for the funding for this work. And that's the end of my presentation. Thank you.
Thank you so much. First of all, it was an amazing presentation, really complimentary to the previous talks, and well under time. So we appreciate that. So we're going to have questions through the chat. I had a couple of questions myself. Let me start with one of those, I guess, for Dr. Anderson, the last speaker. So might I correct that once the hematopoietic cells get into the brain, they're going to be as glia and not necessarily as neurons. Is that correct?
Correct, because we'll be genetically modifying the blood stem cells, which don't in the body turn into neurons. However, there's been papers that you can directly program hematopoietic stem cells into neurons. But yes, they'll be in the glial cells.
And those papers, can they be programmed in vivo into neurons? Or how would that work if you needed to correct a defect in neurons or express a gene in neurons?
Yeah, not with our approach. We'll be using the blood stem cells as sort of a protein factory, which will be constitutively expressing and transferring the receptors. The only thing is that, so our current work on Angelman and other neurodevelopmental disorders, they use regular intracellular proteins like UBE3A is a ubiquitin ligase, and some other ones we're using are transcription factors. However, these GRIN proteins are receptors that are normally expressed on the cell. So we're making certain modifications to hopefully express and then transfer them to neurons, or possibly when there's neurodegeneration, these glia cells actually have direct contact with neurons. So maybe they'll be able to transfer the receptors. So we don't really know the hypothesis, like the direct hypothesis of how it's going to work, but there's a couple mechanisms that potentially could work.
Right, and even in cases where we really think it's a neuron, there are so many cases where we really don't know in terms of a specific disease and pathogenesis. There might be features that are really critical in patients that are those expressed in glia, astroglia, and non-neurons. And some of that we can test preclinically in mouse models. Some of that we don't know until we start using some of these new delivery systems. Okay, so let's see, we have a question from somebody to everybody. But I'll ask that one next. There was a previous question about delivery. And so, clearly there's a balance between the efficiency of gene editing, gene transfer, gene knockdown via, interfering RNAs and delivery. And one question that was already addressed was, the changes, gene editing, do they have to be done more than once? And it was answered in the chat. If you're correcting the genome of a cell, that's for the life of that cell. My question was really for Dr. Kleinstiver. For gene editing, conventionally, at least for many technologies that are available to most people now, you need to deliver something like CRISPR with 2 different viruses. And so the efficiency of getting those both into the same cells in vivo is generally lower than if it was just with one virus, and it might not work if it was so low in the brain. So what's going on with respect to your knowledge to delivery and really getting to be efficient where you're using some of these more complex genetic gene therapy systems where you need to get in more than one molecule into a good number of cells?
Sure. Yeah, I think that's a great question. I think in general, that constraint typically only really applies to delivery by AAV. Most other delivery vehicles have the ability to deliver, a full CRISPR enzyme and the guide in, one construct. I'll highlight the one progeria paper that was actually done using 2 AAVs, where you have to split the Cas9 base editor into 2 pieces and deliver those in separate AAVs. But by using a clinically applicable dose of AAV, not high levels where we see toxicity in patients, they actually saw correction to the point that it actually alleviated the disease in those mice. So I think it really depends on the disease, what level of correction you need, and whether you can deliver sufficient AAV. And then also for some technologies, whether or not we can identify smaller versions of these CRISPR enzymes that can be packaged into a single AAV. So for gene knockout approaches where you just need a nuclease, you don't have to decorate the Cas9 enzyme with these other proteins. That's a lot easier to package in a single AAV. So Editas has been using a single AAV to deliver a small Cas9 enzyme called SA-Cas9 and 2 guide RNAs in a single AAV. There's a lot of effort in the field to work on these smaller Cas enzymes where you can deliver a base editor version of that with a single guide also in a single AAV. So this is definitely on the radar of the entire field of how we can leverage AAV more efficiently for different diseases in vivo by solving that limitation. But I also think it is possible to split these enzymes into 2 AAVs and still have sufficient levels of correction depending on the disease.
Beautiful. That's a great answer. And, you know, it reminds me of we shouldn't, as researchers, be too daunted what we see as restrictions before we start trying them in cellular and animal models, because in some cases we really don't know what the efficiency is, what efficiency is needed for which, you know, disease endpoints in the models. So to that point, and there's been a couple of questions, and I scribbled this question down as well. So Phoebe discussed from working Dr. Ramsey's lab, treating adults. So we all know these GRIN diseases are, for the most part, neurodevelopmental. Whether or not the gene needs to be expressed during childhood development to create all the problems that occur later. In some cases we can presume, in some cases we don't know. But what can we do about, and I'll address this to Phoebe or Dr. Ramsey, what's going on with looking at, at least the mouse models, in the developmental window that's either needed for the development of the disease was required, or what the critical window is for therapy in diseases like a GRIN1 or a GRIN2B or a GRIN anything.
Phoebe, I'll start, and maybe if you have something to add. What I would say is that we've only started to do this with GRIN1, and it's not been done very much for the other GRINs or the GRICs or the GRIAs. So there's a lot of preclinical work that needs to be done. The general wisdom would be that the earlier you intervene, the better it would be. And I think, you know, also the immune system of the infant versus the, you know, an older child, it might be easier to achieve a better level of vector delivery without, you know, kind of the immune system attacking the virus. So there might be several reasons why it will be more successful in an infant, but I would imagine that when the clinical trials are conducted, there will be decisions made about what age will be the target age, and it probably won't be newborn babies, and it probably won't be adults. It will probably be, you know, children, but not necessarily infants. But we'll see. I won't be the one making that decision. So, Phoebe, do you have anything to add on that?
Yeah, I just want to add that I remember when we talked about this project in the very early stage, we did discuss like maybe like later on in this project, we might want to go into an early age to deliver the vector. But as a first study for the gene therapy, we want to like use the dog mice, because we know they can be rescued in adulthood. And the benefit to test if they can be rescued in adulthood is like we can also treat the patients that are like the patients that already have symptoms for years. Like we want to see if the gene therapy could also work for them as well. But later on, we'll definitely want to go early stage and see like if the children can benefit from this or not as well, and like if they can actually get more benefits. So yeah.
Thank you. That's great. One comment and then one more question, and I think we're done with the session. So one comment is in the animal models, there are conditional models that you can either turn on or turn off at different times, and that can be used to define a window. And there's also simply using some of these treatment tools, whether by AAV or a lipid, to really sort of ask, what is the window, either for therapy or for activating the mutation, because you can use a viral CRI to induce a mutation at different times postnatally. So we can use the models we have, both at the viral end and the animal end, to really learn more about, at least in mice, what is the window. And so one more question, and I guess this should get posed to Dr. Anderson. There's been a couple of questions about the immune response and how much stress does it place on the immune system, on the person, to deliver hematopoietic therapies, and sort of an extension of that is, word on the street about AAV that people have been concerned about, in particular, is, eliciting immune response when delivered systemically multiple times. Although I know there's work on the capsid and the vectors and identifying toxic sequences, but would you like to sort of comment on, generally speaking, on delivery systems and how well the patients will be able to handle that in terms of stress or the immune response specifically? And then we'll end the session after this.
Yeah, so as far as stress to the immune system, we always run toxicity assays with any approach. It's required by the FDA. We will be expressing these proteins in all of the hematopoietic system. And so if it works in the mouse model, then we'll move on to the toxicity assays. And so we don't know yet if there's going to be stress with this specific GRIN expression, but we'll be able to test that if it is effective. And then as far as the immune response for multiple gene modifications for our system, we do it ex vivo. It's not a direct injection. So the immune system doesn't see our vector, and then the gene product, so the cells that are gene modified that the patient will get, they actually get tested for residual DNAs that we use for our vector preps. And so we know if any of that VSVG protein is there, which is the vector receptor that we use. And so as far as our approach, we wouldn't see it, and it's potentially a one-time treatment if we're able to get enough of the hematopoietic stem cells gene modified. For AAV, it could be different.
Thank you very much. And again, it's something to worry about, for sure, be aware of, but at least at the research end, not be daunted by that, because people are working on all aspects of this, the toxicity, the exposure of the delivery system, et cetera. So we're a minute or two over more than we were given for the session. So thank you very much, Keith, for not cutting us off. The organizers, thank you very much for the session. I'd like to thank the speakers for great talks, for sticking to the time that we were given. And I want to turn it over to my friend and colleague and collaborator, Ann Perduri, who's going to be leading a family story session. So thanks, everyone, and I look forward to the rest of the meeting.